Hello, this is the next episode of the Paris OG podcast where we learn about entrepreneurs, creators, visionaries, and their pursuit. This episode is a 3-episode podcast focusing on Dr. James and Dr. Howard, who are oncology drug development physicians. They are on a mission to modernize cancer treatment in Dubai and the Middle East at large. The goal is to develop non-invasive, modern, precision cancer treatment. This is the first episode, and we focus on the cancer ecosystem, pharmaceuticals, regulators, and regulation, and what it takes to bring a drug to the market. Enjoy.
Well, first of all, I would like to know more about how this ecosystem works in terms of cancer, oncology, and pharmaceutical in general.
I'll pass to you for that. She'll probably better at explaining the overall ecosystem globally is kind of quite complex.
So the global ecosystem is, I think, to very briefly summarize it, it's Western centric.
Right.
The long established pharmaceutical companies, generally based in Western Europe, North America, and there's to a degree in Japan as well. And drug development is an expensive business. It's become more and more expensive.
Right.
As the technologies have got more complex, and the drugs have been developed for smaller and smaller populations, the regulation around that hasn't evolved very quickly to support that. So the process of drug development is burdensome, it's lengthy. But don't get me wrong, the regulators in those countries are excellent. There's a lot of expertise, a lot of remarkable expertise in those countries. But developing drugs for smaller and smaller populations mean that the drugs that come out of the other end are more and more expensive. So they're developed for and in highly developed healthcare systems, which can leave less developed healthcare systems out in the cold from an economic point of view.
Right.
And then even if the drugs that are developed are made available outside the sort of highly developed healthcare environments, then there isn't necessarily data to support their use in the populations outside those economies.
Well, why is this?
Economics, the finance of the pharmaceutical world. Yeah, it's like, as I described, if we go back 40 years. Cancer treatment was, first of all, it was a lot less effective, but it was also much more simple. Lung cancer was lung cancer, breast cancer was breast cancer.
Right.
Now we know that since the advent of molecular biology in the 90s, we know that every type of cancer is actually dozens or maybe hundreds of different types of cancer. And beyond that, every person's cancer is different. And beyond that, even within an individual, their different tumours within their bodies can be different. Mm-hmm. So, just thinking about oncology specifically, that means that we're developing drugs for smaller populations, but the regulatory process, which itself is built on decades and decades of learning. Digging into the regulations, almost like going on an archaeological dig back through time. That hasn't changed much. There are efforts to change it, make it more efficient, and make it more flexible, but it's still kind of a one-size-fits-all.
Is this on purpose?
No, not as such. No, not on purpose. It's just a function of regulators being public bodies, huge organisations that just don't move very quickly.
There's another function there too, which is that you had, like you were saying, you had very simple treatments to match very simple diagnoses, and actually the treatments weren't very good. Mm-hmm. You had relatively few of them, and your regulators had the time to devote resource to those limited number of available therapies. Mm. You fast forward, and now with the dawn of personalised healthcare and personalised medicine, there is a vast array of highly complex treatments now coming to market, and the regulator is sort of inundated by requests to review this data.
Right.
And they only have the process still that applied to that very simple, very small number of medicines 30, 40 years ago. And playing catch up for them is incredibly hard because the speed of drug development
is very
their ability to evolve. So as a regulator, they face huge challenges creating a framework that's appropriate, and that's kind of meaningfully engages with the pace of development and meaningfully engages with this era of personal medicine, because they've just not got the background, data or the history or the kind of setups to do that. And it wouldn't be possible for them to. How would they?
Right.
And it's not like they don't engage. They're just, because they're large and they're slow, by dint of their size,
the bigger you are, the slower you are.
And they're kind of, they're stuck in the middle of that whole economic cycle pharmaceutical cycle, where you've got the economics of pharma and you've got the pharma industry itself, and the interplay between the three parties, if you will, is very complex, which doesn't necessarily, well, it's a help and a hindrance, obviously. You've got an economic cycle that's volatile, that impacts what the pharmaceutical companies can do.
Right.
That in itself speeds up or slows down the development of certain drugs. Mm-hmm. You've got a regulator sitting in the middle that's trying to determine the best possible rules to protect patient safety. Mm-hmm. That in itself can speed up or slow down the development of certain drugs.
Right.
So it's a potentially frustrating game for the pharmaceutical companies and the investors. And it's probably frustrating being a regulator, seeing people come to you with designs or data that either you're unfamiliar with or has been done in a manner that doesn't really align to best practice as they know it.
Right.
So that, I think, further complicates the field.
Yeah.
Where does that leave the patient?
Well,
That's the big question.
in the
Because
countries-
because when you mention these three people, the regulators, economists, and the investors, and the oncologists or researchers as well. Mm-hmm. If you leave out the patients, it means there is nothing, right? So where does that leave the patient?
Well, in the countries where the drugs are generally developed and that they're developed for to be launched, that although development is in those countries, development is probably slower than it has to be.
Right.
But ultimately, they do get the drugs. It's not ideal. There's a school of thought that says that the current system of drug development, phase 1, 2, 3, marketing authorization, and away you go, that even that's outdated, and you could make drugs more widely available early in that process in more of a graded way. Mm-hmm. So there are ways that things could be speeded up in Europe, in America. But ultimately, patients in those countries should get access to those drugs. Arguably, the bigger problem is that patients outside those countries may not get access to those drugs for a long time, maybe until...
Due to cost or regulation?
Cost, yeah.
Right.
Cost, as I mentioned before, there's also the issue of sort of racial genetic differences as well.
Right.
Sometimes that doesn't matter, but sometimes the safety profile, the efficacy profile might be different in populations outside of Western Europe or North America. For example, if you look at melanoma, one of the most common mutations is a BRAF mutation. The drugs have been developed to target that mutation, but that mutation has a different prevalence in melanoma populations all over the world. But those BRAF mutations are present in about 50% of patients of Western European or North American descent. So fundamentally, drugs are developed for those populations.
Right.
We do bridge them.
Yeah, we do. Yeah.
So, for example, if you do a trial in Western Europe, you can bridge that trial into Japanese patients. Mm-hmm. So what you do is you do clinical trials that compare the early phase data at first between your Caucasian subjects and your Japanese subjects. And then the Japanese regulator will look at that data and go, on the basis of what we're seeing here, this is safe to move into a Japanese patient population.
Yeah.
But it's about the only population that I'm aware of that's routinely being undertaken for.
So do you think if this sort of bridge, is it possible to have this sort of bridge with GCC or like Southeast Asia?
So there's two problems with GCC. Also, so the kind of the region here has two specific problems. One is that historically getting Arabic patients into drug trials has a very low uptake compared to getting Caucasian patients into drug trials. Part of that is obviously the availability of drug trials themselves. So the healthcare economy here having drug trials to offer. Another part of that is the perception or the public perception of drug trials.
Right.
And patients are understandably very adverse to taking something that perhaps isn't fully developed.
Right.
You know, you are exposing yourself to risk by taking part in a drug trial. And as drug developers, we do our best to mitigate that risk, to explain the remaining risks, to support patient safety during those trials. But nevertheless, it's not something that every person, every culture, every region feels entirely comfortable with still.
Cool.
And we want to work to, I guess, to reduce that stigma and to encourage people to access what is potentially their last available treatment option. Certainly in cancer, a lot of the drugs that you and I have worked on over our careers have really become, the last thing that you can take at the end of all of the other treatments. You've exhausted every other treatment option.
Right.
And even then, people can still be reticent, understandably. They're not thinking about necessarily the benefits of that drug.
The upside, yeah.
Well, they're thinking about seeing their family in their
Right.
final months. They're thinking about spending time with their loved ones, doing the things they want to do, and suggesting to them that they commit significant time. And these patients are committing a significant percentage of potentially their remaining life to attending a drug trial, to contributing to this research.
And to society at
And
large.
to society. And it's actually a very big sacrifice for them to come onto a drug trial at that late stage in their cancer. So you have to be respectful of that in terms of your public health messaging and promoting trials. And I think also a very important part of it, certainly in oncology, is making sure that families are on board.
Cool, yeah.
It's not just the cancer patient who's suffering. You've got everyone around them is also suffering in a different way. And I think the good drug trials that I've certainly seen are where we have great clinicians running them who are good at the communication and explaining the time, explaining the risk, explaining the potential benefits to not just the patient but their family. So that's one thing that we're keen to work on in this region. Another thing.
What about the availability of patients?
Interesting. So cancer incidence in this region
It's very low, probably.
is relatively low. So it's about 35% of cancer incidence in Western Europe, for example. They run at around, I think it's 110 to 115 diagnoses per 100,000 people.
Right.
And in the West, that's somewhere in the 300s, low 300s.
So that's a lot.
But that's kind of data as of the last decade. As the kind of steady encroachment of Western diet, certainly,
Right.
makes its impact felt upon the population here within the region, there is an expectation that incidence will steadily increase
Due to
diet, lifestyle, and we're looking at a steady increase from maybe 600,000 deaths a year regionally to over 1 million deaths a year regionally by 2040. So that's quite a such
such a change.
we're on the cusp of that.
Right.
So now is the time to make the interventions or start the interventions.
Preemptive.
Effective.
Yeah.
Yeah, you've got preemption, you've got effective healthcare systems and healthcare policies in place now to tackle that as it occurs.
And actually, cancer incidence and cancer deaths increase as healthcare improves.
Why is that?
Reversely.
Why is that?
Because and this is what's happened in the West, is we've through prevention, through diet, through reducing smoking, and through medical treatment, heart disease, which used to be the biggest killer, has reduced. So as we get better at treating heart disease, people stop dying of heart disease and
Stop dying of cancer.
just live to an older age, where cancer becomes, it's coming up fair to say it's inevitable, but
It's a narrowing
the
gap.
older you get, the higher your chance of cancer, irrespective of how well you've lived your life. How healthy.
Let me ask a question. So, if you live a relatively holistic, healthy life, no smoking, nothing, no hazardous, maybe you live in some very greenish ecosystem, right? Does your chances of getting cancer gets reduced in any way?
It would be reduced, but not to zero.
Not to zero.
Because cancer is almost it's a natural phenomenon. We're all made of billions of cells. As cells divide, that involves replicating the DNA in every single cell, and there are really quite remarkable repair mechanisms to try to prevent cells dividing in an imperfect way. But it's happening billions and billions and billions of times. It just mutations happen, and cancer can just happen. You can live the healthiest life, completely secluded from modern society, and if you live long enough, it's quite possible to likely that you'll get cancer.
So apart from now, so from what I see here is that the more advanced you are, the more likely you are to get cancer.
Or be diagnosed with it.
Or be
diagnosed. Healthcare system.
Right, be diagnosed.
Cancer is more likely to be picked up by a public health run screening program.
Right.
It's more likely to be picked up because you have access to healthcare.
Right.
Simple access to healthcare is one of the biggest impediments, and we see even in places like North America, the geographical size of a country becomes an impediment to access to healthcare in these highly organized healthcare systems, because you don't have a cancer treatment center at every local hospital. What we've done is to improve the quality of cancer treatment, we've concentrated those treatment centers. Now, in a small country like the UAE or a small country like the United Kingdom, that's fine. You're never really that far from a hospital. In bigger healthcare economies, in big countries, or in big countries with smaller, less developed healthcare economies, traveling to a cancer center may become a genuine, traveling even to a hospital becomes an impediment to diagnosis, and then an impediment to treatment.
And also there is the fear of ignorance is bliss, There is this term of ignorance is bliss, where you don't know you have it, it gives you a good feeling.
Yeah.
Or you do know you have it and you don't want to face up to it, which is...
Or you are not sure if you have it or not.
Yeah. And people do prefer to know. People
prefer not to know.
Especially men are appalling for delaying diagnosis. Or
psychological
men are
safety.
much worse at presenting to a doctor with health problems. And it's one of the great public health battles for all physicians, is to persuade patients to present early and to empower them to want to have those conversations with a physician.
Yeah. The best example being prostate cancer.
Yeah, prostate cancer, in men. Absolutely.
Just because you have to have a very sort of personal examination as part of a prostate cancer diagnosis, and a lot of men just bury their head in the sand. There's a big thing in the UK, and I'm sure in other countries as well, to encourage men of a certain age, which I guess I'm reaching now, to be screened for prostate cancer, and it's a little bit taboo. It's more taboo in certain cultures than others. But yeah, prostate cancer is a good example of that. Men bury their head in the sand. They may have symptoms, but they won't seek help for months or even years.
Yeah.
So is there a way we could put some sort of psychological safety before people would want to gear towards this?
I think there's actually quite a good public health campaign in the UK at the moment, encouraging people, it's not specific to prostate cancer, but just encouraging people, if they've got symptoms that they're worried about, to go and get checked up, and the campaign kind of reassures people that 9 times out of 10 it's okay, and it's better to know. I think that'll be that's part of the ecosystem that we're talking about, that we're hoping to develop. Your screening would inevitably be part of that, but it is hard, because it takes a cultural mindset change.
And investment too.
And investment, yeah.
So the great thing about the UAE is that you have a government that's very proactive about supporting initiatives to improve quality of life, to improve anything really. If it's possible to improve it, there's an ear that will listen here. So I think the major thing is investments in public health campaigns like we have in the UK. I mean, the campaigns in the UK have been interesting. You've got posters on bus stops, radio, television. There's a very broad-based campaign. And I think the other thing is obviously with a semi-privatized healthcare system such as that which exists in the UAE, it's important that the government works to coordinate public health messaging across that private sector.
Right.
It's quite easy in the UK because the majority of our healthcare economy is public in the form of the NHS. public health messaging is disseminated in a coordinated manner. That's harder to do when your hospitals are privately owned and managed, but it's not impossible. And I think most clinicians obviously are very committed to improving the health of their patients and improving the health of society overall. It's the majority reason why they go into being a doctor. So actually the government supporting those private healthcare, communities and hospitals to enable their clinicians to spread a coordinated public health message is probably super important. And I think it then that follows into things like getting patients to understand the importance of clinical trials.
Right.
Developing a clinical trial infrastructure here will require the government to sit down with experts and to come up with a coordinated message that helps not only develop that economy, but helps improve patient health and patient access to care as a result of that.
Oh, well, that's very promising, I think. So, about regulators. Let's assume you're a regulator. What would you love to change?
I think it's important to say that
What would you do differently?
it's not regulators. It's not a negative relationship with regulators. Pharmaceutical industry and regulators do work very, very closely together because they have.
You need each other, right?
Yeah, absolutely. They're an essential part of that economy, and you shouldn't be doing things without regulation.
Without
because
regulation.
it leads to patient harm.
Yeah.
I think what we would seek to change, certainly within the personalized healthcare cancer space, is the speed with which you can develop drugs based on the same technology or same platform, if you will.
Yeah.
So, for example, what we're looking to do involves effectively activating the body's own immune system against cancer by taking cancer cells within the body that have been hidden and highlighting them to the immune system. That's basically what the kind of drugs that we develop have been focused on doing. The trouble is, obviously, as we say, because there is this genetic component to personalized medicine, there is the potential to need to develop drugs that have slightly different targeting molecules to treat the same cancer, but in different patients. Now, the overall structure of the rest of the drug can remain the same.
So it's like a bigger system where you can take smaller parts.
It's like a little bolt. You have a bolt on where you just change one end of the drug slightly. And you're not changing what it's trying to do, you're changing what it's binding to.
And you're not changing the whole component.
No. You're just changing the bit which binds.
Yeah.
And especially in this region, there's quite a variety of binding sites that potentially might be required.
Right.
It's slightly easier. Caucasian populations tend to have a very preserved binding site, for want of a better word, and it's quite easy to make a drug that targets a large
population. Right.
Yes. Here, the play might be slightly different in that you would hope that the regulators would allow you to provide a robust preclinical package. So that's a package of data before you've tested it on humans, where we would look at all the different binding sites using artificial intelligence, so what we call in silico testing on a microchip.
Right.
And then,
so like when you say microchip, like something that will...
No, actually on a computer.
Oh, okay. When you say microchip, a lot of people is like my brain on my body, and they're like, woo!
It's this new frontier of kind of preclinical testing where we can use computers to simulate the environment in the body and what drugs might do to that environment.
Oh, cool.
So not only can you use computers now to find drug targets, so you take patient data, genetic data, you give the computer that data, and it can actually help you narrow down a field to then go and test in animal studies or laboratory studies with tissue, so tissue samples.
Yeah.
So no humans are exposed. But you can then, you can use it to then speed up that development process in humans because you've done a lot of the work beforehand and proven that the multiple targets you're looking to bind to have the same safety profile, hopefully. At the moment, you cannot do that. It's very difficult to do that. You would have to run separate drug trials, separate preclinical
a lot of money,
packages,
a lot of
separate
investment.
drug submissions. And this is where this investment part of the ecosystem really kicks in. You're asking investors to hand over billions of dollars per drug potentially now.
For something you don't know if they're going to have proof.
And for something that targets a smaller number of people because it's personalized.
Right.
And you've got to persuade them that it's worth continuing to invest in that over time. And they do have to see a return on that investment. I think in pharma in general, it's a bit of a dirty word to talk about profit. It can't be a dirty word to talk about profit. We have to look upon it as an essential part of the ecosystem,
Right?
Because it's what funds making more drugs.
Yeah.
And if regulators can get on board with a change in the way we approach early drug development preclinically, to allow us to test similar, drugs across the same platform without going through that process every time, it's much more likely that you're going to garner investment. It's much more likely that you can make drugs quicker that save people's, save or improve people's lives, and that you can use that return on investment to develop more drugs. So I think that's the major thing for us is condensing that platform submission, or the submission of a platform of drugs into a single submission. That's done all at the same time, with a single associated cost. Because drug development costs are rather fixed overall. It doesn't matter what drug you develop; it costs roughly the same amount of money. And obviously, when you're targeting a smaller group of patients, you've got to make that money work a bit harder.
So to kind of draw that in a different way, if we're talking specifically about T-cell receptor targeted drugs, and I won't go into the detail of that. James kind of explained it very well. But the target, the thing on the tumour that you're targeting in different people is displayed slightly differently. So for most drugs, if you develop that drug, it could potentially be used in anyone in the world with that cancer, or anyone in the world with that cancer with that mutation. That isn't the case with T-cell receptor targeted drugs. If you wanted to develop a T-cell receptor drug to hit that target on that cancer, you would probably have to have, I don't know, I'm guessing 20 different drugs. It's probably in the dozens to hit the global population.
You could probably do 70 to 80% of the global population with 5 different target molecules.
And then there's diminishing return.
But it's a diminishing return after that, because you've got hundreds of potential different surface targets on that cancer cell. Also, and I think it's maybe worth a small explanation, is that what's on the surface of that cancer cell is a fixed presenting molecule, which is the bit that changes in different people, and then a little portion of that cancer, a tiny little fragment of that cancer's protein, which sits in that presenting molecule in essence. And you can make drugs that bind with really, really high affinity to that complex. The trouble is when the presenting molecule changes, it won't bind anymore. And this is what we're trying to impact with a change in regulatory environment, is that you can make one that targets 5 of these different presenting molecules, or you can make 5 drugs that target 5 different presenting molecules, but all target the same little fragment of the cancer, without having to do multiple submissions, because the cost is just enormous. I think the other thing to say there is that the reason for wanting to do these medicines that target this complex, because I suppose the obvious question is, why are you making such a complex drug? Why make something that's so targeted that it has to work on a specific profile? It's really because you've got for increased longevity, the potential for reduced symptoms because they're so targeted. But if people don't develop these drugs, and we were talking about this yesterday, if you don't develop these drugs, you're never going to find the thing that makes a step change in cancer therapy. You're never going to find the magic bullet in cancer, which I suppose is a horrible term really from a clinical point of view. But if people aren't continuing to do research, if people aren't continuing to make more specific drugs, we're never going to find a combination that leads either to a cure or a functional. Because at the moment, most cancer therapy is still staged in a very traditional manner. You might have radiotherapy, so radiation therapy. You might have chemotherapy, so drugs that are toxic to cells. You might have surgical resection, where they go in and take out part of the tumor to make it smaller. But those are technologies that have been around for 50, 60 years.
So there is nothing new right now.
There's plenty of new things, but there's still the kind of core treatment that people being diagnosed with cancer still receive. Once they've moved through all of those, and sadly their cancers continue to progress, or has come back, or has grown, Then you start to look at more targeted therapies, such as the ones that we're developing. And there are a number of them on the market that are very good.
So hold on. So the ones you're trying to develop right now, is it less invasive or not?
They're more targeted.
More targeted.
So hopefully, and this is generally the case, the side effects are fewer. Because chemotherapy is a very blunt instrument.
It is there. It's like you're killing the person until the cancer dies.
You kill all,
for the
you're
person.
just killing rapidly dividing cells, which covers cancer, but it covers a lot of the other bits of your body as well. So you're killing all those cells, and people get really sick, and the chemotherapy can kill people.
Yeah.
So the kind of immunotherapy we're interested in and we would like to develop for the regional population is more targeted, and they are potentially more efficacious as well, particularly when used in combination. So T cells are your body's own natural cancer defense. Even as two young men and one older man sitting here now, our T cells are constantly checking every cell in our body to see if anything has gone wrong. And if they find that something's gone wrong, they will kill that cell. That surveillance is constant and going. So T cells are naturally very good at killing cancer cells, but cancer can evade that surveillance.
That's surveillance, right?
And the T cell receptor targeted drugs just kind of switch on again that ability for T cells to recognize cancer cells. So using that process and almost certainly in combination with other immunotherapies or other targeted therapies, and also doing that on a personalized level where you are targeting an individual patient's mutations and accounting for their genetic type, as we've been talking about. Everyone's different, their HLA type is different, and that's different between different ethnicities. That's where the hope is in the future for possibly cure, certainly possibly longer survival with cancer. But as James says, to get there, we need to keep innovating, developing the new drugs, which under the current system always end up at the end of the line. That's where drugs are developed generally to be used last line. New drugs come in last, because you do the trials there because you can test against best supportive care against nothing, essentially. So it's a relatively low bar, and then they move forward, which takes a lot of time, and that's something else that we're good to address.
You're not exposing patients to the risk of an untested drug compared to something that's known.
Yeah.
So it'd be very rare to run a test where you said, well, you've just been diagnosed with cancer, we'll give you this experimental drug, or you'd have the traditional therapy. We have the data that supports the traditional therapy, and it takes a very long time to build up the same confidence in the data and the same amount of data for a novel therapy. So that's not something I think we can necessarily expect to impact, because I think that if you made regulators amenable to bringing forward drug trials to sort of early diagnosis, it would be very potentially risky. There are cases where you can do it in combination. There are cases where the safety profile of a drug in trial would support that, but it's not common. And it's not an approach that most physicians are hugely supportive of for obvious reasons.
Yeah.
Like you've got to really present very compelling data. Normally, you present that compelling data at the point at which your drug is already marketed, and it's something that drug manufacturers obviously have a commercial benefit from, is to bring forward the stage of therapy that they can use their drug at. Because the earlier you can bring that drug forward, the more people will have access to it, the more people you can sell it to, and the quicker you can return.
You can beat the competition as well.
And you beat the competition, of course. And there is that commercial element. We all want to make the drug that cures cancer with like that.
And probably most importantly, the earlier in treatment you use a therapy, the better the outcomes.
Yeah.
It's pretty much it is a universal law. The returns are lower as you move down lines of treatment. So
why is this? Because the price comes down, or what?
Cancer's better at evading.
The longer a patient has cancer, the more mutations, the more escape mechanisms it's already developed, and just the sicker the patient is. So you may develop a great new drug that's used as monotherapy, that's used on its own, and maybe third, fourth, fifth line. and that may improve survival by 3 or 6 months. If it's used eventually, first line, possibly in combination with something else, it could add potentially years to life or a year to life. The same drug used earlier will give you much better results, but new drugs are almost always developed last line.
Because of the safety
Right.
and cost issue.
Yeah.
So I've seen situations where a patient was treated with a cancer treatment, the cancer was gone, and then the same cancer came back.
So I guess the misnomer there is the cancer probably never went.
Yeah.
The cancer just became so small.
Dormant.
Not even necessarily dormant, just, yes, sometimes dormant, but
Yeah.
so small it wasn't detectable.
Yeah.
And this is the problem. We don't have imaging or diagnostic tests that are going to detect every last cancer cell in your body. So we currently, on the whole, use kind of advanced imaging and relatively advanced guidelines for reviewing that imaging.
Yeah.
Which are globally standardized.
Right.
So radiographers, who are the people who take the scans, and radiologists are the people who review the scans, have very specific guidelines for cancer, which are rules as to how you read a scan and how you read that tumor. But the imaging can only show you
What it
certain
sees.
degree of
Yeah.
acuity. Like, you can't see individual cells on the. There are scans that will do that, but they're not available commercially. Where you can't see tumors at a cellular level on those scans. Now, one of the I was going to touch on this. So, what do you want to touch on it?
I believe you're talking ctDNA.
Well, ctDNA and also like stained
And PET.
specific like stained imaging or targeted imaging.
It is.
Which we can't touch on. You'll have to listen this bit out. We can't touch on the one we were working on.
Oh, not
commercially.
specifically, no.
That's NDA, but like,
Yeah.
it's worth the discussion.
Yeah. So what James described there is a traditional way of monitoring cancer and cancer treatment, and it's still the standard in a lot of cancers, and it depends where you are. But we talk about using MRI or CT scans to see if you can actually see the tumor, and if you've been treated, has that tumor vanished to the resolution of that scan. But that doesn't mean that every single cell has gone, has been eradicated, and that's why cancer comes back. You can't see to a cellular level on a CT scan or an MRI, but it's a huge area of interest now, and there are different ways of scanning where you can see to a high resolution, like PET scanning and things, where you can you will almost make those cells that are remaining kind of glow, so it's possible to see them.
Right.
But probably the most exciting technology there, which has been utilised more and more in certainly in drug development and in clinical practice as well, is using ctDNA, where you will get a blood sample from a patient, and you can. So cancer inherently has mutated DNA. That's kind of what cancer is. And you can detect that mutated DNA in patients' blood, sometimes even if you can't see a tumour. And that's been used more and more to direct treatments. If a patient's had whatever it might be, surgical treatment, radiotherapy, medical treatment early on in their treatment course, if after that treatment they still have cancer ctDNA in their blood, then that can prompt further treatment.
So this should be a compulsory thing to do? Since
Probably
it's
probably
possible.
not at the moment. So I think it's worth mentioning. ctDNA stands for circulating tumor DNA.
Yeah.
Right.
So kind of complicated acronym for a simple thing. It is standard in medicine, but there isn't. It's like anything. There isn't enough data at the moment to say that this is absolutely going to be our new gold standard.
Yeah, the gold standard, yeah.
Kind of diagnostic or prognostic test for cancer. It is something we're using alongside the advanced imaging, where we're using PET scans where you tag the tumor and it glows. You can use other molecules where, depending on the type of tumor you have, you can bind specific things to it that allow you to visualize it on scan better. And that's another emerging field. But it's the combination of all of these imaging and diagnostic techniques together that at the moment can help to guide treatment at a physician level. So there's not a national body at the moment, or there's not a kind of industry body that is, or a regulatory body that at the moment is saying you must do ctDNA testing. what there is a growing body of evidence within the pharmaceutical industry and within clinical practice that would support generating guidelines for that. And I think that is something that you and I would be very keen to see.
Yeah.
See us reach a data threshold where we can have the confidence in it to say this is now what we're going to use as gold standard.
Right.
Yes, we still need the imaging, because it is important to know if a tumor is going to impinge on things, to know if there's a possibility for surgical resection of that tumor based on its location, because those are potentially powerful things to help a patient's cancer journey, to improve their health.
Cancer journey.
Well, it can still, it is a journey.
It's a journey, yeah. This is a horrible journey, yeah.
But you know, so there's still a need for imaging. There's still a need for high quality, standardized, rigorously reviewed imaging. But that shouldn't be the point at which we as physicians go, we don't need anything else.
Right.
And what we talk about here is, just to clarify, is solid tumours, not blood cancer. In blood cancer, ctDNA is used routinely, and it has been for a while. Solid tumours, it's a relatively new thing, and that's what James has described.
Detecting it was not the easiest. You're talking about a tiny circulating fragment of DNA from a tumour that you've got to separate from a normal blood sample, which in and of itself is an amazing thing. You're actually providing a diagnostic and potentially prognostic test from a normal blood sample, which actually, you know, you're not exposing the patient to any real risk there. So that's fantastic. You're not having to go into the tumour and take a sample of it, which historically is quite a risky procedure depending on the location of the tumour, size of the tumour, how we use the term friable, but basically cancers, because they're mutated, the cells don't bind together very well. So actually, if you go and start poking around them with a big needle, you run the risk of potentially spreading that cancer because a piece could break up. You run the risk of patients bleeding really badly because even the blood vessels in a cancer are often not very strong. They can leak very easily.
Yeah.
So in and of itself, just having that test there is a massive benefit to patients already in terms of them being able to come into a standard health clinic even, just have a blood sample and it gets sent off to a laboratory, and they're not having to be admitted to hospital, yet another procedure for that, you know, because of their cancer diagnosis. So I think not just ctDNA, but non-invasive testing and the support for non-invasive testing is something we'd be keen to see more of. Because anything that minimizes the burden of treatment to a cancer patient is something we should promote.
Right.
If it is a high quality test, and if it can provide high quality outcomes that clinicians can act upon.
So this is probably a stupid question. You mentioned T cells acting as surveillance. It's like a sentinel in the body. So is there a way we can make them more surveillant? Like sometimes, because you mentioned that cancer cells can evade T cells. So is there a way to make more T cells or make them more surveillance?
It's like CAR T, isn't it?
Well, actually, if you think about more surveillant, that's actually what immune checkpoint inhibitors do. So the big step change in IO, in immuno-oncology, cancer immunotherapy, happened about 15 years ago now, when immune checkpoint inhibitors were trialed in melanoma, in skin cancer. And this is fascinating, but I'll try to describe it simply. T cells are constantly surveying for cancer, as well as for viral infections, but we'll stick with cancer, are surveying for cancer. If they become activated, so they recognize something wrong with a cell, over time, your own body dampens down that reaction as kind of a self-preservation.
Does your body get used to T cells afterwards?
No,
or?
you'd get very sick. Imagine having the flu. You'd constantly be generating an almost flu-like response, which is what we commonly see with these immune therapies, is that activating your body's own immune system is the reason when you get flu, you feel ill. It's not the influenza, it's not the flu. It's your body going, I must fight this.
I must fight this, and then you raise your temperature, right?
I'm going to shiver. I'm going to do all these things that make the environment hostile, along with attacking that actual virus, or in this case, the cancer. So it's why children, when they get sick, often have a very high fever, then they're better quickly. Whereas adults, because we've got a more acutely tuned, a more fine-tuned immune system, and we're better at the surveillance when we become adults, tend to mount a much more aggressive response and get quite unwell with it. And it's the reason people always say, children just bounce back when they get, minor illnesses. They don't have very good surveillance. They're still developing that surveillance, and it takes probably until early adulthood, in truth, to finish developing that surveillance system to its peak. After which it's fine until you're kind of, I guess, 50, 65 years of age, and then it slowly gets a little bit less good every year after that, in very crude terms. But sorry, I interrupted your checkpoint.
So when, let's say a cancer's developing, T cells are in that tumour, the cancer will, partly naturally, but also as part of its altered mutational state, will express checkpoint inhibitors. And that basically stops the T cells doing their job.
Right.
So it expresses checkpoint molecules, which stops the T cells doing their job. We can now give, and these are successful in lots of cancers now, initially melanoma, but lots of cancers. We can give checkpoint inhibitors that take the brakes off the T cells again and let them do their job. So in a way that improves the surveillance. One of the downsides of that is you remove the checkpoint activity all over the body. So the side effects of those drugs are autoimmune disease, because T cells can start attacking your own tissues, whereas previously they've been stopped from doing that. But that's a way that we have addressed, I think, what you asked, a way of improving T cell surveillance. But I don't know, your question might have asked, can we improve T cell surveillance before cancer even develops? Just, sort of make us superhuman. Now that isn't...
It's like when you have a wash tower.
Yeah.
It washes right, but if you increase the wash tower to 2, 3, 4, more security.
Probably not until the point you've got a mutation, for the simple reason that, or it's probably not until you've got an illness, because the way the immune system works is really quite exquisite in that if it finds something that initially looks not that correct.
Not that correct.
And it will have a very general response to that. The longer that...
So it doesn't think.
Well, no, it doesn't think, but then it starts to think. That's the thing. If that insult, or if that mutation, or if that thing that's not quite right sits there for any length of time, your body's immune system starts to try and generate a more targeted response to it. This is all, again, in very high-level terms, but in essence, you refine the T cell. The T cells develop a better response, or a more targeted response to the thing that they don't like. And that works in cancer, it works in viral illnesses, it works in bacterial illnesses. Your immune system will evolve to try and treat that specific insult in the best manner possible. And there are cancer therapies out there, like CAR-T, where you are modifying, you take cells out of a patient's body, you modify those T cells to be more specific for the cancer. You genetically type that cancer, and you find out exactly what mutations it has, and you will try and put those cells back into that patient's body, having customized them effectively. And this is, again, extremely high level.
Yeah.
The problem with those therapies is they do have quite aggressive side effects.
Yeah, it's the conditioning as well.
Sick.
You need to give chemotherapy before the CAR T cells to kind of half destroy a patient's immune system, so they
don't fight.
So they will adopt the transplanted cells. It's an amazing technology, and I think the way it's going to develop over the next decades will be fascinating to watch. At the moment, it's they can be effective, particularly in blood cancers.
Yeah.
But they're not without their downsides at the moment because of the conditioning. But largely because of the conditioning regimes.
And actually the reaction to re-exposure, I guess.
Yeah.
When you actually put the cells back in, people can get quite sick in response to that. So, but that's not just, this is, I think, a common misnomer is that even when we say that the drugs that we're looking to develop have a more tolerable side effect profile, patients still require surveillance. They're still going to require, certainly in the early stage of their treatments, and the first month of treatment often, when they're coming in, they're going to need to stay in hospital for a little bit of time, not days, but potentially overnight, from a safety point of view, because we know that if they're going to have a really strong immune reaction, that can make them feel a bit sick. And that's the kind of most common, I would say, side effect profile of these drugs. Obviously, with any drugs, there's the risk of them upsetting the liver, upsetting the kidneys, and that normally can just be monitored. But there's always the potential for people to get really sick with that, and you have to then keep them in hospital to manage them. It's rare, but I think the idea that people can come in, have a cancer therapy, and walk out. It's an event horizon we're looking towards always. Right. We always want that drug where you give it to, you give someone a pill and
walk away.
Home. It would be very foolish of us as physicians, I think, to believe that that's something that's likely to happen anytime soon. And it would be even more foolish to try and subject patients to a trial regimen that attempted to achieve that. The patient safety is always more important than whether or not the drug is convenient to take. And again, that's another thing where when you're counseling patients about drug trials, giving them an accurate expectation is super important to that physician-patient relationship. Building trust by giving them honest, accurate information, not trying to sugarcoat it, is very important. And in actual fact, where physicians give a poor understanding to their patients of what a drug's going to be like, what a drug trial will be like, you overwhelmingly end up with that patient choosing to leave the trial. It almost always results in the patient withdrawing not only themselves from the treatment, but their data from the trial. Whereas actually, patients who are really well counseled, understand really well what they're going to go through, tend to stick the course. Or at the very least, they're very amenable to having a discussion with you about staying on. Maybe you make some accommodations for them, especially in cancer trials. We tend to be quite accommodating. We tend to write the trial in such a way that if patients are sick for a week or two, we understand that they can come back on trial if at all possible, which in a normal drug trial you wouldn't. Non-cancer drug trials, patients not attending, you would just remove their data from the trial. Cancer trials, and I think it's worth highlighting this, do tend to be written with the needs of those patients in mind. We know that sometimes, for example, I used to see this a lot, patients want to go on holiday. They want a final holiday with their family. It's super important.
They've been dreaming about it for a decade.
And they've gone, I never got round to doing it. I'm going to do it now.
No, you never know what's tomorrow, right?
And you'll try and accommodate that where possible. To the point, you'll move dosing around a bit. You'll give them a treatment break for a week if they're established on therapy. So I think it's part of that public health message is that it's not this kind of faceless machine where you're being ordered and told what to do. Physicians will work with patients to absolutely the utmost of their ability to make that treatment or that drug trial tolerable, and to keep patients engaged, to keep patients compliant, to get the really good data that we need to make these therapies work for patients once they're marketed. But I think that's a big fear people have. I think people look upon drug trials as maybe like you're going to put in almost like a prison. Like you're going to be locked in a hospital ward for weeks at a time, and you're going to be told what to do.
They're going to be washed and stuff like that.
And yes, there are things where you'll have to follow rules, but on the whole, especially in cancer, we try and make them as friendly and accessible, and to the point where obviously when we're developing trials, you develop a global network of specialists, so people can visit a local cancer center or even have therapy with their local oncologist.
It takes a lot.
They're not going to have resources necessarily to a new doctor.
It takes a lot of resources.
A lot of resources, but actually keeping patients in contact with their existing oncology doctor, or having a situation where their trial oncology doctor is communicating with their local oncologist routinely, so that everyone's kept in the loop and knows what's happening is super important to us.
Wow.
So the improving T cell surveillance tends to be, because of the side effects that we talked about, it is reserved for when people have cancer. At the moment, although I think it's a really interesting concept. I haven't actually thought about it. It's something to think about. I don't know if any work.
There could be a breakthrough,
Give us a decade.
I can't wait, honestly.